Okay, can you hear me? Yes, okay, great. So this is my second time coming to this wonderful conference, and I had to tell you, I hate public speaking, but when they invited me back, I couldn't say no, because I really love meeting so many of you. I think this is such a great opportunity to really build a community of people who are really focused on PLS and HSP. And I could not emphasize enough how important it is to meet and advocate for your own disease. As you've just heard, there's many different rare diseases, and obviously we want HSP and PLS to be up there front and center so that we can advocate for more research specifically for your conditions. So I'm here as an advocate for PLS. I've been lucky to receive a grant from your foundation so that I could work on PLS for 2 years, as I will explain in a second. So I work in Boston at the Massachusetts General Hospital, and I do research, clinical trials, and clinical research, both on ALS and PLS, again, thanks to your support. So I'm here because I was funded by something called the Virginia Freer Sweeney Fellowship. I'm actually the second fellow. The first fellow is here with us, Dr. Christina Fournier, and she'll speak right after me. And again, I cannot emphasize enough how important it is that your foundation sponsors people who are gonna really support and promote research in PLS. Because again, there's not much research out there on PLS specifically because it's a rare disease. And what you want is to create a community of researchers who can really promote and really move us a step closer to finding a cure for PLS. So Christina started a few years ago. She did 2 years of fellowship, and then I'm the second fellow. When I came last year here to this conference for the first time, I had just started. So I just presented my plans. Today, that's my first real progress report for all of you. I hope you'll be pleased with some of the progress we have made. I still have a few months left in the fellowship, so I'll finalize the results by the end of the year. Everything will be published in scientific journals, and obviously I want all of you to have access to the data as soon as it becomes available. The final publication becomes available. So I'll explain a few projects and a few goals of my fellowship. First of all, when I started, obviously I wanted to improve our knowledge of PLS. A major goal was also to discover new PLS disease mechanisms, and all of this with the goal of promoting PLS trials. And again, I cannot emphasize enough, and I want to say that once more, that it's important to create a community and an infrastructure, just like we heard in the previous talk. It's important to have an infrastructure and groups of researchers that really want to promote research on your specific disease. So I'll start by discussing what we have done over the last year and a half to improve our knowledge of PLS. So I'm gonna talk specifically about the Neals PLS registry. So one more acronym for you. Neals stands for the Northeast ALS Consortium, which is a network of clinical centers, academic centers, hospitals that do research specifically on ALS and PLS. This was started in the 90s, so it's about 20 years old. Started in the Northeast, as the name says, and the map shows the active locations. It's about 120 sites across the country. Again, most in the Northeast, but now covering the majority of the country. And it's a network, it's an infrastructure of institutions that can run ALS clinical trials efficiently. So we leveraged this infrastructure to identify and do research on people with PLS, because people with PLS oftentimes are seen in ALS clinics, because PLS is somewhat related to ALS, although it's a different disease, as you know, with different progression. But oftentimes people are seen in ALS clinics, so it was a natural partner for us, again, to partner with Neals, because we knew that many people with PLS are actually seen in Neals centers. So we could go to those centers and identify people with PLS. And actually, Christina Fournier, who will speak next, when she was the first fellow in PLS, she started a registry, a cross-sectional registry on PLS, and then I expanded that as part of my fellowship. So as you know, PLS is very rare. There's many rare diseases. PLS is particularly rare because it's only about 1,000 people in the US. That's an estimate. Just by comparison, HSP has about 20,000 people in the US with HSP. So PLS is really rare, and ALS is also about 20,000 people. So PLS is a small proportion of that. So how do you find those 1,000 people? Well, previous studies were trying to locate people with PLS, and on average, they would study 10 to 50 patients at a time. Now, we just heard that CREATE is gonna expand that. For our registry, the Neals registry that we are gonna speak about today, we identified about 275 patients, which is a good proportion of the PLS population in the US. Now, we want to emphasize that different registries, it's very good that there are different registries because each registry has different goals. So the fact that there are parallel studies, we CREATE or this registry or other efforts, it's actually quite good because these different registries are complementary and they serve different goals. So you can ask different questions and get different answers, all complementary and synergistic towards finding a cure. So specifically, for this registry, this was a retrospective registry, as I will explain in a second. And so it's a little bit different from the CREATE registry that you just heard about. The CREATE registry has a large biomarker component as well. This was more focused on understanding clinical patterns of progression of PLS. So where do these patients come from? Well, the patients that were included in this registry came from 21 academic centers, 13 states in the US, and some contribution from 2 academic partners from Canada and Israel. So again, that's a large percentage of the PLS population in the US. So when you do a registry, you can set it up in different ways. Let's say that I want to know what's happening today, how many people with PLS there are, and what's happening to them today. There will be a cross-sectional registry, and actually Christina did that when she was the first fellow. She published a paper describing, again, the distribution of people with PLS and some of their characteristics. And I think that was very important because when you have such a rare disease, you really need to go out and proactively reach out to academic centers to identify patients and also sort of, promote some interest in the research of this rare disease. So based on that first project, now we knew where PLS patients were seen at Neals sites across the country. And so we began to create something called an upper motor neuron task force at Neals. And actually Frank Davis came last year and attended that annual meeting, and I think he has reported that on your newsletter. So it's very important basically to have a collaboration between the Spastic Paraplegia Foundation and this committee and Neals so that the two can work together to really reach out to people who see PLS in their clinical practice and again, promote their participation in research. So again, when you look at somebody today, you can do a cross-sectional registry. What I did for this particular registry was something different called the Retrospective registry. So I only had 2 years to do that because the fellowship is only 2 years long. So when we were thinking about this, the way was, we were thinking, well, how can I maximize my time? How can I, really look at data longitudinally, meaning over time? But I only had 2 years of time to do that. So we looked back in time, and we did a retrospective registry. What that means is that we asked several people across Neals to give us data from electronic medical records about the last 15 years of history of their PLS patients. So basically, we followed all these patients, kind of looking back in time, from the time they were diagnosed with PLS, and then we asked investigators what was happening to them over time. And again, just as you have heard, it's very powerful to know what the natural expected progression of a disease is, what we call natural history. And the reason that's important is because the more we know about what's expected, what would normally happen, the better equipped we are to see if a treatment, such as exercise or a drug or any other intervention, is actually changing the natural progression of the disease. So it's very important to really lay the foundation for clinical trials. So that's what we did over the last couple of years. When I presented here last year, I told you that we had designed all the forms, we had designed the database, and we were beginning data collection, and now I'm happy to report that data collection has been done. It's finished, so we have all the data from almost 300 patients with PLS. All the data has been entered in our electronic medical records kind of database, and now I'm going to spend the last few months of my fellowship analyzing the data so that I can tell you more about things like, are there more males or females? What's their age of onset? What's their pattern of disease? How do they progress over time? And again, that's by looking at their history, sort of looking at their medical records. And for this particular registry, because again, it was a retrospective registry, we didn't need patient contact. So actually, it's very possible that some of your data may be it's completely de-identified for privacy reasons, so we don't know who that belongs to. But we can really do a lot of research and learn the natural history of PLS based on this registry. Now, the most powerful type of registry is something called prospective registry, something like the CREATE registry, where you start today and keep following patients over time into the future. So you can collect biomarkers, for example, biosamples, and you can ask them questions over time. Well, I didn't have time to do that during my fellowship because that was only 2 years long. And as you can imagine, if you want to have a long follow-up time, you need a lot of time to accumulate data in the context of a prospective registry, and that's also more expensive. But I had the good fortune of collaborating with Dr. Mary Kay Floater, whom many of you know. She's spoken here in the past, and she has actually started another prospective PLS registry at the NIH. She started in 2000. She has been following about 80 patients with PLS on an annual basis since then. And she was kind enough to share some of the data with me. So we haven't published that yet. We're still analyzing some of the data. But the idea was to, again, learn about the disease as it progresses over time. She sees patients on an annual basis. Some of you may have actually been meeting with her at NIH. And she does a complex battery of tests. So every year, people undergo a few tests to really try to dive a little bit deeper into their progression and how they present. One simple questionnaire that I just wanted to discuss today that we have been looking at is called the ALS-FRSR scale. It was actually mentioned in the previous talk. The scale was developed for ALS, but can be applied to PLS as well. And so basically it asks people how they function in their daily life. So for example, how is their speech, or how do they walk? And it tracks people over time to see how that changes over time. So we looked at the data from these 80 people, and we graphed the way ALS-FRSR progresses over time in PLS. So the good news is that, not surprisingly, the way this declines over time is actually slower than in ALS. And why is this important? Well, when you are trying to develop a sensitive scale, something that's sensitive to change, you want to develop a scale that's good enough, sensitive enough for you to see change. In other words, if then you apply a drug, for example, that may have some therapeutic effect, you want to be able to see a change on that scale. But if the change changes very slowly, that's harder to do. You need more people in your clinical trials in order to be able to detect a change. So what, a next step for this would be to develop actually a PLS-specific scale. So we have heard before there are some spastic paraplegia scales, and a new scale that's been developed now, thanks to actually your contribution from this organization as well as a private donor, is a PLS-specific scale. And that's led by Dr. Hiroshi Mitsumoto, who also spoke here in the past. He's from Colombia, and is leading the PLS-specific scale. That's sort of a different version. It starts out from the ALS-FRSR, which is well validated, but now it's going to be more granular. It's going to ask more detailed questions to people with PLS specifically. And the hope there is that the PLS-specific scale is going to be more sensitive to change. In other words, it's going to be better to be used in the context of a clinical trial to detect a change or an effect of a drug. And many of us, at Neals are part of this effort, and so we are asking our patients with PLS to be part of this so that we can validate a new scale that's going to be PLS specific. So why am I telling all of this? I told you that my first goal was to better understand PLS. So we've done the registry, we're doing new scales. Why is all of this important? Well, that's important because all this work lays the foundation for clinical trials. In other words, we all want to ultimately develop new drugs. But if we don't know about the disease, if we don't have a scale to measure the success of a drug, then we're set up for failure, basically. So instead, with all of this work, we're laying the foundation for clinical trials where we can be well positioned to see a treatment effect or a positive effect of the drug. So I told you that the second goal of my fellowship was actually to discover new PLS disease mechanisms. And that's what I'm going to talk about next. So first of all, just as a summary of what happens in PLS, we know that there's essentially one cell type that's mostly affected in PLS. The nerve cells that are affected in PLS are called upper motor neurons. They are the same cells that are also affected in HSP. And they're located in the brain, and then they send sort of signals through some cables called axons to the spinal cord. And ultimately, that allows for movement. So if these upper motor neurons are not working well, obviously, movement is going to be affected. So we wanted to look at what happens in the area surrounding the upper motor neurons to see if there was anything that was essentially disrupting their ability to function. So just to make an analogy here, I want to use the analogy of the telephone game. So as you know, in the telephone game, somebody speaks on one end, and then the signal is transmitted through a cable to the end of the line. And then if there's more outside noise, the message gets distorted. So again, that's pretty much what we wanted to see. Is there anything that's affecting the way upper motor neuron cells are working so that the message doesn't get transmitted properly to the spinal cord? So how can we study that? Well, at our institution, we have a machine that's called an MR PET. Other institutions have that too, but the unique feature of our system is that they are combined. Essentially, the PET scanner is built inside an MRI machine. And what that helps with is basically it saves time, and also it's very more comfortable for the patient because you basically can get more information in a shorter amount of time. So what do you do with this? Well, with the MR portion of it, we're going to look at structure, or you can think of it as the hardware. So you can look at the upper motor neuron cells and their cables as they go from the brain to the spinal cord. And then with the PET portion of it, you can look at function. You can think of it as the software. So how are these cells working together to produce movements? With PET, you have to inject a dye or a tracer to look at some specific function. There's different types of function that you can look at. So what we are doing is to use a tracer called PBR28 that binds to some cell types in the brain that regulate inflammation or neuroinflammation. So we are trying to see if there's inflammation surrounding the nerve cells, the upper motor neurons. So again, we're looking at the upper motor neurons. We're doing brain MR PET to look at sort of that noise that's going around the nerve cells. So this is what we saw in PLS. So the arrow points to the motor region of PLS, and the red signals means that there is increased neuroinflammation around the upper motor neurons. And this tells you what it will look like in somebody who does not have PLS. So green is good, and red means more inflammation. And I just want to point out, this is not the type of regular inflammation that you can see in the body. I'm not talking of an autoimmune disease. I'm not talking of inflammation that you can treat with anti-inflammatories or steroids. That's a different type of inflammation. I'm talking of specifically brain inflammation that's located in the brain. So we need to figure out perhaps a way to modulate that. So we also looked at how that relates to structural integrity, meaning how much the cables what is the structure of those cables that send the signal. And so we looked at some MRI measures. And here you can see the orange circles are people with PLS, and the blue circles are people who don't have PLS. And we saw more inflammation in the motor regions. And that also correlates, the more inflammation, the less structural integrity you would see in those cables that send signals to the spinal cord. And then you can also look at the actual cell bodies, the cells themselves. Are you going to lose cells over time? And we did that, again, with MRI studies. And the blue stripes are the motor regions, where you can see in people with PLS, the more blue this is, the less cells you have. So you can see the loss of the nerve cells that overlaps with the areas of inflammation. So why is this important? So why are we looking at hardware and software here? And actually, I would say, I want to add that many of you actually participated in this study, so thank you very much, because obviously all of this would not be possible without you. But as we develop, more knowledge about new disease mechanisms, we can use this knowledge to promote PLS trials, which is ultimately our goal. Now, we don't have a PLS trial based on these results yet, but I just want to illustrate sort of the framework that we're using to potentially develop new drug trials with these techniques. So one thing that we could do if we had a drug that targets neuroinflammation would be to do proof of mechanism studies. In other words, let's say that you scan people with PLS that have red signal, and then you give them drugs in different types of drug, and you see the ones that turn the signal from red to green, as you can see on the right. Then we would know that that drug has engaged its target. It's doing something in the brain. So last year when I spoke again, I had this sort of concept in mind, and I'm glad to report that now we are doing exactly this in ALS. It's not PLS yet, but it's actually quite important to see that this concept is actually gaining traction, and it's actually happening in ALS. And then obviously the more we know about PLS, the closer we will be to doing something similar for PLS as well. So now we have 3 drugs that we are testing in proof of mechanism trials in ALS using this specific technique. These are the names. They're not for PLS, again, they're for ALS. And then hopefully we hope to select the one that works the best to bring to larger trials, which will be efficacy trials. So that's one possibility. Another possibility, another application of this technique is to move towards something that we call personalized medicine, which is basically the concept of one size does not fit all. So how would you do that? So you can imagine that you have a rare population. We don't want to waste your time. We just want to select the people that are most appropriate for this type of drug. So we scan these people, we identify the ones that have the most neuroinflammation, and we give a drug that targets that mechanism to those people, so to that cohort, a specific group of people, and perhaps other people are less likely to respond, so we wouldn't give that drug. Again, these are just concepts, but I just want to convey the fact that in ALS, this is a concept that's actually gaining traction. We're actually moving forward with trials following this kind of methods, so hopefully the more we know about this type of mechanisms in PLS, the sooner we will have trials for PLS as well. Now, you may wonder, when will we have a drug trial for PLS? And actually, I'm glad to report that there is one active trial that's enrolling people with PLS for a drug that actually has a different mechanism, nothing to do with neuroinflammation. That's a trial of Ampera in PLS. It's, again, proof of concept. This drug is used in multiple sclerosis to help with gait. So the goal of the drug is not to change the natural history of the disease. It's not to change the way the disease is going to progress, but to see if it can give a little bit easier ability to walk to people who take it. We don't know if it works, it's just a trial. And that's led by Dave Lange and Jim Weimer, so it's available in New York, Florida, and also at our site in Boston. So again, some progress, again, we don't know if this is gonna work, but it's exciting to see that there is more interest in PLS to the point that there's actually one enrolling trial at this time. So again, I want to conclude by saying that as we learn more about PLS, as we discover new mechanisms, we can move closer to clinical trials, and most importantly, we can create a community of PLS researchers. As you know, funding is limited. There's many different rare diseases. You want to advocate for your disease. You want that your disease to be front and center, and that's what we're doing at Neals. We are trying to, again, promote an interest on PLS specifically. And again, we have an annual meeting with Christina and other people at Neals trying to continue to promote interest on this rare disease. And my mentors for this are Mary Sukovic and Nazem Atassi. We continue to work together with the Neals team. And again, our goal is to support your mission of finding a cure for PLS. Thank you very much. Okay. So that's an excellent question. No, that's not the last step. So when right, that's a great question. So when you do clinical trials, you have different phases. You have phase 1, phase 2, phase 3, and that's a good thing. What I mean, I'll give you just a little summary. When you do phase 1 and 2 studies, they are safety and proof of mechanism studies. So that's the first step. You do it in a small group of people because you cannot do a phase 3 study right away. A phase 3 study is something to test really the efficacy and get approval from the FDA to use the drug in the entire population. But those are much larger trials. So we cannot test every single drug in the context of a phase 3 study because that requires hundreds of people. And so again, especially with a rare population, you want to really maximize your phase 1 and phase 2 studies where you have a small group of people and you can be very efficient in screening new drugs, and then you select only the most promising drugs to do a larger study for FDA approval. Now, why am I saying that? Because you want to make your phase 1 and phase 2 studies as efficient as possible. So things like imaging would be considered a biomarker or a proof of target engagement for that one that you are studying. Now, it depends on the mechanism of the drug. So if the drug targets neuroinflammation, you can use PET that measures it. If the drug targets something else, for example, Ampira, the drug I was talking about, may have some electrophysiological properties. So there is some electrophysiological measurements that are done in the context of that trial. So again, the idea is to gather as much data as possible that tells you which drug is the most promising. So this would be something to maximize the impact of early phase trials. And then you would select those drugs that are the most promising to do a larger phase 3 trial. Does that answer your question?
Yeah, the measurement then would be just to see the efficacy of the drug once you try it.
Well, in order to see the efficacy and really gain FDA approval, you need to also show the efficacy on clinically meaningful results. So basically, you need to see really an improvement in clinical function. So an imaging or a biomarker would not give you that necessarily. So you also need to make sure in the context of a large phase 3 trial that the drug really changes the natural progression of the disease. There you still have to do that to prove efficacy and to get approval. But again, keep in mind that those studies to show efficacy, clinical efficacy, are very large, long, and expensive. So you cannot think, it would not be feasible to test all drugs with that concept. And also, I would say for PLS specifically, we don't even have great clinical measures. So part of the work in terms of doing registries, really developing new scales, functional scales, questionnaires that can really give us clinical information. That's quite important because as of right now, even if we had a great drug, it's hard to measure the effect in PLS unless you have a very robust scale. So all of these efforts, whether they come from our registry, CREATE, or other efforts, they're really important to really provide the foundation for really designing the best trials.
Lots of variables that take a long time.
It does. It does. Exactly, yeah. And I would say that, your organization is really critical in really promoting PLS and really promoting the fact that we can do research on this disease. So as you can imagine, when you only have 1,000 patients around, most researchers may be discouraged. So by having infrastructures such as our consortium or the consortium that was mentioned before, we can really create the infrastructure to do this type of studies. Unfortunately, it has taken time, but now we have the infrastructure. So I think that this is the right time to invest money and resources on doing PLS research.
Thank you.
Yes.
Could you just explain the concept of neuroinflammation?
Right, that's actually a little bit of still a controversial concept, but the idea is that there are cells in the brain that are not nerve cells. In other words, the brain is made up of both neurons and supportive cells called glia. There are some types of glial cells that react to inflammation, which is a good, react to damage, which is a good thing in a sense. It helps, for example, fight infection or other problems. But at the same time, when they are abnormally activated in the context of some diseases, there is some evidence showing that they can actually be detrimental. So it's like a double-edged sword, right? So they fight inflammation, damage, or other things, but at the same time, they can be activated in a way that's abnormal, that actually creates or perpetuates the damage to the neurons. So it's tricky how to modulate that. It's not well known yet. And again, I think we're still sort of trying to figure that out. There are animal models in ALS where it's been clearly shown that this type of neuroinflammation can actually be detrimental at certain stages of the disease. So again, we are trying to figure it out in PLS. Now in PLS, we don't have an animal model, I should say. We don't have a mouse that replicates PLS. So it's a little bit trickier to find cause and effect, because in people, we cannot do those kind of experiments. But this is as close as we can get, I guess, in PLS. And we're learning from ALS, from the animal models of ALS.